Okay,
Thank you.
so let's get started. Thank you very much, and thank you. I would like to thank the organizers for giving me the opportunity to speak here. So before I start, I need to declare a conflict of interest. I'm co-founder of Nautisha Biotechnologies company. So we know that gut microbiome is a very complex ecosystem, and it's also very important because it's almost like an essential organ and provides many important functions, as the previous speaker already showed you. Now the question is, how can we identify the most important members which are providing such essential functions from such a complex system? And first, we need to do the so-called genome resolved metagenomics. We should do deep metagenomic sequencing, and then we should assemble high-quality genomes of the major members of the gut microbiota. And when we get the de novo assembled genomes, we should realize that bacteria are not independent from each other, and they actually interact with each other and form a functional group called guild. So guild is a concept in macroecology. Different members of a complex ecosystem, such as rainforests, can actually work together to form a coherent functional group as a guild. So members in the same guild, they increase, decrease together. They show co-abundant behavior because somehow they work together and to become advantageous in competing with others. And now the question is, how can we identify guild-level organization among the genomes? And a very important hypothesis for identifying important bacteria as guild is that if different bacteria form a guild, and that guild also very important in providing essential health relevant functions to the host, then the members in the guild will need to keep a very robust ecological relationship with each other. Even when you change the microbiome and their abundance are changed profoundly, their relationship should be kept stable. So this is our hypothesis. And based on this hypothesis, when we try to analyze our genome resolved data, we should look at search for stable genome pairs. So in this published type 2 diabetes clinical trial, we already showed that high fiber diet can significantly change the gut microbiota of type 2 diabetes patients and also alleviate disease. So we did another large scale clinical trial. So we randomized patients, type 2 diabetes patients, into either high fiber diet group or control group, and gave them a high fiber diet in the control treatment group for 3 months. After that, we stopped the intervention, and then after a year, we get everybody back and have a medical checkup and analyze their microbiome again. So for this sample set, we assembled 1,845 high-quality genomes as the variables for microbiome analysis. So you can see when you give high-fiber diet, and the gut microbiota changed from here to here, and very dramatically. But after you stop the high-fiber diet, it eventually go back to the same region. This is for the high-fiber treatment group. Well, for the control group, during the 3 time points, there is no change of the gut microbiota. If you look at the disease, HbA1c significantly reduced after 3 months on high-fiber diet in the treatment group, but it bounced back after you stop the high-fiber treatment a year, and disease come back, microbiota went back like a baseline, and disease also came back. So we selected 477 genomes, which are shared by more than 75% of the samples at each of the 3 time points in the treatment group. So we constructed a co-abundance network across all the individuals at the same time point. So we have the M0, M3, M15 co-abundance network. And then we search for genome pairs which can keep stable ecological relationship at M3, M0, M3, and M15. So this means that if two genomes, they always have a stable ecological relationship, no matter how much you change their abundance, that means they are very stable genome pairs, and there is a reason for them to become such a strong genome pairs. So eventually, we identified about 600 such genome pair relationship, and they involve 141 genomes. These 141 genomes actually organize into two guilds, and this guild 50 genomes, and this guild 91 genomes. And interestingly, all the genomes in these two guilds, they have at least one very stable relationship with another genome. So they are the most stably connected genomes. And they are also the most highly connected, because this 141 genome already takes up more than 85% of all the connections among genomes. And if you look at the genes, guild 1 hardly had any detrimental genes. Only one genome had antibiotic resistant gene, and 3 genomes had one single virulence gene. Well, the genomes in guild 2 mainly had all different types of antibiotic resistant gene, and also have all various types of virulence genes. And guild 1 had more genes for degrading plant polysaccharides, and also more genes for producing butyrate, and also acetate. So this indicated that genetically guild 1 can respond to high fiber diet, and also potentially beneficial, while guild 2 actually potentially pathogenic. And interestingly, during the study, these two guilds has a seesaw relationship. So guild 1 increase, guild 2 decrease, guild 1 decrease, guild 2 increase. Well, disease would go away or come back. And by using these 141 genomes as variables to construct a machine learning regression model, we actually can predict 41 out of all the 43 clinical parameters. And is this an inherent ecological pattern of gut microbiota, or it's just a transient structure and induced by the high fiber diet we used? So we tested this by using these 141 genomes as reference, and we found that in another independent type 2 diabetes case control study, metagenomically studied, both the healthy control and the disease cohort showed the similar two competing guilds structure. And also by using these 141 genomes, we can actually classify disease from control successfully. And then we collected data from from 12 other independent metagenomic study, case control study. So atherosclerosis, liver cirrhosis, ankylosing spondylitis, and also Parkinson's disease, schizophrenia, colorectal cancer, IBD, and hypertension. Not only different diseases, but also the cohorts are from various continents and different ethnic groups. So this 141 genomes, by using this 141 genomes as reference genomes, we can actually detect the variations of such genomes in all these independent data sets. And by using this as a variable to do a machine learning classification model, we can tell disease from control successfully from all the data sets. So this is actually a highly conserved core genome, a core microbiome in human gut microbiota potentially. And maybe evolutionarily selected by the high fiber diet ancient human have been using until recently we use a low fiber diet. But for millions of years, humans are using high fiber diet. So such a structure may have been evolutionarily selected and also conserved. And so because of the time constraint, I would just stop here and hope I can answer your questions. Thank you.